You're good?
It's okay on screen.
Yeah, looks good.
Time the. Oh, cool. Okay. Hi everyone. My name is Landon. I work in the corporate genetic lab at UCSC. I'm an undergrad there. Today I'm going to talk to you about a systematic search for introner elements. So before I get into introner elements, I just want to talk a little bit about spliceosomal introns. I'm sure most of you are familiar with spliceosomal introns and know that they're spliced from mature mRNA transcripts, and also know that they're a hallmark of eukaryotic genomes. And spliceosomal introns are known to play important roles in genome structure evolution, or thought to, but we don't really know too well where they originated. So there were originally two theories that sought to kind of describe how introns came to be. One of them was intron early theory, in which the last universal common ancestor contained introns, and introns were slowly removed from prokaryotic genomes and are slowly being removed currently from eukaryotic genomes. In contrast, intron late theory suggests that the last universal common ancestor did not contain introns, and that introns invaded eukaryotic genomes after the divergence between prokaryotes and eukaryotes. And along with that invasion came the evolution of the spliceosome. So there are a lot of proposed mechanisms for intron gain. These are just a few that I've listed up here: intronization, intron transfer, genomic duplication, intron gain during double-stranded break repair. But the one that we're really interested in is transposition. And the reason we're interested in transposition is because it's the only one of these that's really been shown to create mass intron gain on genomic scales throughout literature. So how does transposition cause intron gain? Well, you have an intron that also could be a transposon, and it basically gets cut out of the genome and inserts randomly somewhere else in the genome. And if that occurs, if that insertion site is in fact inside an exon, a coding region, that intron can then insert there, forming a new intron, and then forming out of one exon, two exons. And this can happen on massive scales, where one specific intron could exhibit a burst event in which it transposes all across the genome and causes mass intron gain. So that kind of brings me to introner elements, which are essentially transposable introns. And they were originally discovered in a algal species, Micromonas pusilla, and are described in a science paper published in 2009 by Warden et al. And then were discovered also in a pretty distant relative of Epicilla, NFA jefferens, and that was originally described by Huff et al. In the Nature paper published in 2016. And here I'm basically showing you two sequence motifs. Both are consensus sequences for a particular family of introner elements in one of these species. The top is a consensus in NFA jefferens, and the bottom is a consensus in Micromonas pusilla. And you can notice the GT or GC 5' splice sites pretty highly conserved, and the 3' AG splice site is pretty highly conserved. And one thing I want to point out is that the 5' splice site in NFA jefferens is co-opted from the insertion site, and the 3' splice site is actually carried within the transposon. And the opposite is the case for Micromonas pusilla. So since these are transposons, if there are recent insertion events, they tend to have highly similar sequence similarity, but also length similarity. So here's a couple distributions. The first is in Micromonas pusilla. It's a comparison of the distribution of intron lengths with the distribution of introner element lengths in the genome. And you can see there's a pretty clear difference in those distributions, where you have a particular family of introner elements that there's a very large count at that specific length. You see the same with NFA jefferens. Now, so after kind of reading this paper, I was pretty interested in, I was just curious. I mean, we were curious do these exist in other genomes? And if so, what proportion of intron gain do they account for? So to kind of test this and try to find an answer, we started developing a pipeline in which we do a systematic search on 1,894 assemblies that we obtained through the Genomic and RefSync databases. Here I'm just showing you a tree, basically, of the phylogenetic spectrum of species that we actually we search through. And we only consider species that have a valid annotation because we need to be able to find introns. So that's why that number is much smaller than the size of these actual big databases. But we consider a pretty broad phylogenetic spectrum, where since these were only discovered in algospecies or protospecies before, it's just we wanted to consider all species. So we start by extracting the introns from the genome using the annotation file and the FASTA file. And we move to doing an all-by-all blast between all the introns in the genome to look for sequence similarity. And then we use a graph theory algorithm to cluster introns based on sequence similarity. What you're seeing here is kind of a visualization of what one of these graphs might look like for a particular cluster of introner elements. Each one of those dots, which is a node in graph theory, is one intron, and then the lines that are connecting each of those dots is the sequence similarity between those two introns. And so I'm putting out kind of the line there. So we kind of initiated this part of the pipeline and found a whole bunch of putative introner element families throughout this broad phylogenetic spectrum, but then we had to QC our data. So one of the main things we were worried about is getting false positives from gene duplications. So we removed possible false positives from gene duplications by comparing the exonic regions that flank each intron to each other, and if those are above a certain similarity, we throw those introns out of our data set. And really the most integral part of this pipeline, which is highlighted in different colors because we're not quite finished with it yet, is comparing orthologous genes between species to really demonstrate that intron gain events occur. And that's something we haven't finished yet. It's the most important part of this pipeline, but we're pretty confident right now that we have some intron gain events. So basically here I'm showing kind of the number of species we considered. We're zooming out of that tree that I displayed earlier and kind of classifying everything in that tree under 5 specific broad clades. We have plants, protists, animals, fungi, and then whatever doesn't fall under those 4. And you can kind of see that we found a lot of putative introner elements if you compare that to the number of species we considered in each of these clades, but these numbers are definitely much larger than the actual, the real events that have occurred. We know we're going to continue to weed out false positives as we continue through this kind of QC process and this filtering process, but we are confident that about some of these. So next time I want to show you what introner elements might look like and some of the examples that we've discovered. This is in Plasmodium, Chabadi. This is an introner element family in that species, and you can see between introns in this family, again, there's high sequence similarity. This is a multiple sequence alignment, and you see the 5' GT splice site and the 3' AG. And then one thing I wanted to point out is that if you remember how I pointed out that a lot of these introner elements can co-opt specific splice sites, we have a truncation event in which this specific introner element has actually co-opted a 5' splice site downstream from the original splice site. And then another example is in a fly species. This is another introner element family where we observe pretty high sequence similarity, but this time we observed an elongation event where an alternate 3' splice site has been co-opted. Now, we have a lot of other examples that I haven't displayed up here, MSAs for each of these, and we've observed a lot more differences in sequence length in particular introner element families that have been observed before, and we think that's probably because there's a lot more co-option going on of splice sites. So this is kind of the same cartoon I showed you before, but when this introner element family gets inserted here, it causes an intron gain event, but actually co-opts a downstream 5' splice or 3' splice site and elongates as a result. So next, I just kind of want to show how these introner elements can affect the genomic landscape. We're comparing the distribution of intron lengths across genomes. On the left, we have genomes that contain active introner elements, and on the right, we have genomes that do not contain active introner elements. And you can see that overall, the number of introns in these genomes differ. The ones containing active introners have more introns than the ones that don't. But I kind of want to point out a little bit more specifically where the length of the consensus sequence or the consensus sequence for the introner element families in each of these species lies. And you can kind of see that the distribution is kind of molded around that specific length. And when we think about how these introner elements can co-opt splice sites, and we think about how they could possibly change in length, we would kind of expect to see this kind of distribution around that specific introner element consensus length. So we're really excited to see that. And each of these species are so Microctinium conductrix is a close relative of the other species displayed on the right, and these are both Plasmodium species that are very closely related. So this is exciting to see. So basically, in conclusion, I would like to say that introner elements might operate in many more species than previously thought, and across a much broader phylogenetic spectrum, since there hasn't really been a systematic search performed previous on, they haven't considered all phylogenies. We are excited to find so many putative introner element families where they haven't been discovered before. Also, transposons may generate introns on genomic scales in a subset of lineages by co-opting pre-existing splice sites or encoding their own. And this is something we want to look into a lot further. It's possible that some of these events we're finding are actually transposons inserting into exons and co-opting splice sites, and may not contain either splice site within their actual sequence. And we're really excited about that. But that's slightly different than introner elements. And transposition may account for a significant proportion of intron gain in eukaryotes, but I can't really, we don't have strong evidence for that until we actually finish the last part of our pipeline. We aren't able to give a proportion or a percentage of intron gain events that we can account to transposition. So basically, future directions, finishing that last part of the pipeline, and further examining the structure of introners and regions surrounding them. And this is something I actually I really am interested in. Huff et al., that paper, they explained that a lot of these introner elements exhibited target site duplications around their insertion sites, and I really would like to look for those systematically throughout our data sets and see how that maybe suggests other mechanisms. And also we want to look at the secondary structure. We've done some RNA fold predictions of secondary structures on our data, and have found some promising secondary structures. And we want to know how that might play a role in the mechanism. Happy birthday to my PI, Russell, and thanks to everyone else in the Corbett Lab. It's been awesome, and I'm really excited to keep working on this project. Thank you. How much time I have for questions, but if anyone has one. Yeah.
So introns can have important, big, and highly conserved regulatory elements, but across the genome, across the biology. So I'm wondering how you're planning on filtering those from your data.
Yeah, I mean, it's another QC stuff that we have to get to. It's going to be a long process of filtering data, that's for sure. I think, yeah. Once we figure out if we can detect basically, if we look between orthologous genes in different species, and we can detect their being intron position in one gene where there's no intron in that position in another gene in a different species, then we kind of can recognize that as an intron gain, and I think that's kind of what we're talking about. Hopefully, yeah. Thanks a lot.